So here we have a gel electrophoresis tray, and you can see that it's got a chemical in it that will allow the movement of electricity through it. Basically, you've got power pack, lid, and then what we've got here is the samples. So these are the different samples. So it tells you tube A is a standard dye marker, so that will have all of the different colours. We've got tube B, which is mother 1, tube C, mother 2, tube D, boy 1, tube E, boy 2, and then the father. Now, so what you need to do is just tap it down like that. Just tap it down so you've got all the samples at the bottom of the wells. You have your tray here with the agar jelly in it, and you can see the wells there. You can see the wells there where you add the fragments of your DNA samples. Okay. We also have sterile tips. These are sterile pipette tips, and we're going to be using today an Edvotech micro pipette. I've set it, okay, at 15 microliters. We pick up in a sterile environment, okay, a pipette tip. You only use the pipette tip once, okay? And what you need to do is you need to get the sample. So you would, I'm not going to do it, but you would pierce the sample, okay? But before you pierce it, you need to put the button completely down, okay? And as you put it down into the well, into where the DNA fragment is, and then lift, okay? When you lift it, you'll take the chemical up through the DNA fragment through the pipette tip. You would then very carefully, okay, you would then very carefully press down very lightly to remove the DNA fragment into the DNA well, okay? And to get it all out, you would press the button. When you've finished, you can then, rather than touching the sample, because you could contaminate the sample, all you do is press this button here. There we go. Okay, so you load up your welds, okay, and then you bring it over here, and we're going to place it, and I'm not going to place it at the moment, place it into the tray, and then we're going to connect up the electrophoresis kit and leave it for about 30 minutes. Okay, so the students have put the different DNA fragments into the wells. Okay, and then we're going to place it into the tray. So very carefully lower it down flat, and there we go. So what we've got to do now is we've lowered them down. You can see they're lowered into the liquid, and we're now going to attach the electrical contacts. So negative on that side, positive on this side. Because DNA is negative, it will move to the positive, okay? So we then need to just plug it in to the power pack, and we turn on the power pack, okay? So hopefully over time we'll be able to see things starting to separate. Okay, so we've left it for about 20 odd minutes, and the samples are now separated. So we need to make sure we turn off the electrical power. Okay, so just turn that off, and then remove the top. Okay. So what we can see from each well is a separation of the DNA fragments. Now, luckily, this is coloured dye. So coloured dye with different size fragments. In reality, we would use a fluorescent label to show up the different things, or we could even use X-rays as well in certain techniques, other techniques. And you're going to look at the banding for each of the samples, and you're trying to find out who the boys are related to. Are they related to mother 1, mother 2, and which boy which father, which of the boys has their father that's alive in this sample.